there, you didn't come there. I said, I was just. Any of you have taken any bioinformatics course and have doing anything before? This is the first time? But I remember, I think I saw, because I keep looking at, some instructors come and they offer courses, and I've seen something similar offered by somebody else about bioinformatics in MIT person last year or so. They didn't have any other course about bioinformatics or...
That Dr. Riaz has offered in previous years.
Which one? What was the title of the course?
It was computational and bioinformatics.
Computational. Yeah, this one saying I have seen somebody, offering almost similar course. Did you take that one?
Yes.
Well, did you take my previous one? No. So maybe today you'll find it similar to what he offered. Dr. Riaz is doing research here, faculty here?
Yes, Dr. Riaz.
Oh, Dr. Riaz. Dr. Riaz offered it. Dr. Riaz is he's doing some sort of, I think, molecular imaging in his research or something.
Molecular dominance.
No. Other than that, he's also doing some imaging studies. What happened? Seeds? You also have, this is almost sitting area, right? On the back and on the sides. Yeah, they can. That is all I mean. There are still students outside? How many of you are doing MPhil? Can you please raise your hand? Everybody? Almost everybody? And how many are doing PhD? But now it is mandatory for you to do MPhil before you do PhD?
Yes.
So nobody can directly enroll in PhD? You can put your hand down. So you cannot directly enroll in PhD anymore? Why? What is the difference between MPhil and PhD? Because MPhil is a degree that does not exist internationally, right? And I don't know whether it is recognized or not. But what is the difference between MPhil and PhD?
I want to know what's MPhil. I was asking everyone what is MPhil.
You're asking everyone?
I don't know. First time I asked for.
Where are you from?
I'm from New Zealand,
But
I'm Iraqi.
But what are you doing here? Like you're doing a project?
I'm doctor. I am on the twice fellowship.
No, but then you're doing a research project or you're a sabbatical or what?
Research project.
A research project.
Yeah, I'm a twice fellow.
No, but then you are yourself working on a project with somebody, with another faculty or so, right? So yeah, MPhil, actually I also want to know what is the difference between MPhil and PhD. But my understanding was, when I was here, that in some institutions, because in fact, this was my understanding, I don't know, I might be wrong at that time, it's very, the PhD, it takes a huge amount of years, sometimes 3, 4, 5, 6 years. And sometimes people are not able to complete all the way, 7, 8 years. So in that case, they have offered a break to the students who want to finish after 2, 3 years for some reason. They want to discontinue, they don't want to continue anymore. But the work they have done in the last 2 or 3 years, they don't want that work to be wasted. So in that case, they give them this intermediate degree as MPhil. Then in case in future they want to continue to go through 2 or 3 years of their life they spend in doing MPhil, research work, will not be wasted. It will be counted towards their final PhD degree. This was my understanding. Right? But there were reasons for offering this option. It is in those cases where it is taking extraordinarily long to complete your PhD. But now I find that they have made it mandatory. Why they have made it mandatory? Is it the same thing? My understanding is the same for every
See, your bachelor is not equal to international.
Because your bachelor is not, that's what they are asking you to No, but my understanding also is that when we were studying here, in 4 years you were getting master's degree. But now I think they have made it 4 years you get undergrad degree, right? And then 2 years additional. But in that case, MPhil doesn't make any sense.
It should be
If somebody would be doing MPhil, it is B because we did not do masters, right? But then you guys already have it takes 6 years to complete your masters, right? But it doesn't make any sense for MPhil then.
I completed 2 masters, Master of Science and Master of
Undergrad is 4 years.
Sorry, what?
And BS undergrad is 4 years.
Yeah, that's what I'm saying. And masters is 2 years. It's on top of the undergrad.
No. We don't. I have not done the masters. I have done the undergrad, 16 years graduation, and then MPhil. And now
No, but you okay? Don't quote me individual examples. I just want to know on the whole, like what is like the issue? Like what? Yes, go ahead.
BS 4 years program and BSc Honors 3 years program and MSc 1 year program both are equal. Then after MSc, like the student who got the degree of BS program, also their degree is equivalent to MSc. Here in Pakistan, after that they are eligible for MPhil.
No, I understand how you do it. Like, I know you can do undergrad and masters and MPhil and PhD. My question is why MPhil degree is free? You're not doing masters, but she said you did the MSc.
in Karachi,
if we have an MSc.
No, so it means that somebody who is doing MPhil, they are not supposed to do masters. Like from undergrad directly you can enroll in MPhil. That's how it works. So, but then again, you can directly enroll in PhD. Why in MPhil?
HEC requirement.
it's 18 years.
Yeah. I think you know what my understanding is, but they want to keep you in school for longer. Because I give you an example back in Canada, previously it used to take 2 years to get a nursing degree to become a nurse, right? 2 years. But then they find out by the time the nurses are graduated, they are in the workforce, their jobs not available for them. So what they decided to keep them in the school for longer. Instead of 2 years, now they're taking 4 years to give you the degree. So they're keeping you in the school longer, so that by the time you're graduated, then the jobs are available, and then there is no problem with the workforce availability and the jobs are in the market. So this I understand there, but here, I don't know if they're keeping you for doing MPhil placement, they have increased. How many years it takes now to do PhD? 4 to 5 and BS plus MPhil? Okay, how much time it takes for MPhil? 2 to 3 years? 3 to 4 years? So 4 years of MPhil and then 4 years of PhD? 2 years of MPhil? Minimum. And maximum? 3 years. 2 to 3 years. And then PhD? PhD how many years? 5, 6? Indefinite? Minimum 3, like 3 to 5 years. Because altogether 5 PhD plus 3 and 8 years. So you guys, by the time you are done graduation, you are very old, right? So we were the younger girls when we did our PhD. In our time, I think by the age of 24, 25, we are done with PhD. So I did my undergrad, and from the undergrad, I directly enrolled into the PhD. And PhD takes 4 years or so, right? So by 23, 24, you should be done with PhD.
Supervisor also.
So I don't believe because I dictated my thesis myself. I did not get instructions from my professor. Who was my supervisor? You know that? Dr. Dowman. But our immediate supervisor is Dr. Raj. He did not control anything in my PhD. I decided when I wanted to do my PhD, when I want to finish. I did all my experiments myself. I designed my PhD work myself. And I told him that tomorrow I want to defend. He said, okay, go ahead. So it doesn't depend on the professor, it's up to you. If you are motivated enough, if you are energetic enough, you can convince your supervisor, you have experiments, you have data, nobody is going to stop. Right? Huh? That's why you should choose wisely. Because you are working with the professors and you are going to use their resources. So always choose professors who are rich with resources, who have plenty of expertise available, who have expert seniors available in the lab who can guide you, and who have access to the funds, grants, and infrastructure. Yeah, I think that there are many changes now. I don't know whether it is still working. That I think now the director decides which professor you should work with, right? These are the issues. I don't know, is it still the case?
Yes. Board decided.
But now we have a change in the leadership. Is still the same rules they follow?
No, ma'am.
We can ask
Okay. But now you can also decide if you want to work with something
we can ask
different.
to change the supervisor.
You can change your supervisor?
Ask to change the supervisor.
Yes, it depends upon the supervisor.
I see. Okay, should we start? It's 10:05. We have learned enough about MPhil and PhD. So you know what MPhil is? No, not yet.
No, it doesn't make a sense.
Doesn't make sense in which respect?
It's in my country. I have my PhD from Iraq. I was on private program. Like I finished 3 years PSc and then 2 years master and then second year I enrolled in PhD. I was 25 PhD.
How many years you took to do PhD?
I've done it in 3 years.
In 3 years. No, but immediately you done your PhD, you have jobs available?
No, immediately I done PhD. I enrolled in my department as a lecturer and then I.
So you see, that's the difference. Here they don't have access to the jobs as such. There's a
That's why
saturation in the jobs.
they keep them MPhil, master.
Yeah, that's what I'm saying. They're keeping in the school longer, so that by the time they graduated, then the job should be available. So the problem is not. I don't think so.
But that's
Everybody.
wasting the
Huh?
young generation.
It is frustration. Yes.
Yeah, it's
very frustrated, is it, guys?
Yeah, it's wasting. No, it's waste for a young generation.
It's a time wastage. But see, for them, it doesn't matter. See, majority of here are girls. If they're boys, they will be making rules for them. This unfortunate reality. I'm not just joking about it, but this is unfortunate reality. And the majority of the boys, I can tell you, they are not from the local area, Karachi.
No, they are.
They are all coming from all other parts of the country.
I noticed that.
Yeah, this is unfortunate reality. Somebody has to step in and do something about it. But again, what they can do, because the jobs are not there. Right? And it's not the case here in Pakistan, I'm telling you, it's the same as in Canada also. You won't believe that in my university, the 60% faculties are sessional faculties. Sessional means temporary faculties. So if they get their job from semester to semester. And now, because our university, all over we have recession, Canada is not an exception. My university has a $13 million shortfall. And because of this, they have put freeze on hiring. They are, if they don't have enough enrollment in certain courses, they are discontinuing those courses. They are not hiring the sessionals. We cannot hire sessionals anymore. The ones which were already there, they're just completing their courses and then they are done, right? So this is an issue everywhere. I remember that one of the profs I was talking to, she's working there for so long. She said when she was doing her masters, at that time there were so many opportunities available that she ended up her masters by teaching 5 proper courses to PhD students. Because there was a need for the instructor. There were not enough instructors when she was doing masters. And now it's the time that people do not get their first faculty position until they pass 15 to 20 years of their PhD. For 15 to 20 years, they are struggling. Over there, you have to have a postdoc before you get a faculty position. And initially, the 5 years, 1 postdoc was enough. Now people are coming with 2 and 3 postdocs. So for 15 years, you are struggling after you done your PhD before you get the job. Here, at least you are lucky. We don't have to struggle for that long, right? You find jobs somewhere here and there. Okay, should we start? Introduction to bioinformatics. Why you guys wanted to learn about bioinformatics? Are you planning to start a new position somewhere where you will be talking about bioinformatics, computational modeling, or imaging, or AI, or designing some algorithms?
My research topic is
Your research topic is bioinformatics? With whom you are working? Dr. Riaz?
Yes.
Okay. But you already have somebody available to guide you. Other people, what is their interest? Why are too many people interested in this course? Yes.
I have also computational research.
Yeah, but your supervisor is like, this is your field of research. But I don't think so everybody is doing research in
I have
bioinformatics.
some experiment field and some computational field here, so I choose.
Yes, but other people, they are all from bioinformatics field. Are you guys doing research?
No, I'm biotechnology. I have a project and I need to have background on bioinformatics, so I know how can I predict something. I have hypothesis. I want to predict something in the future, and I want bioinformatics to help me in that regarding the protein, the genome, or something.
What about you guys? Just doing it because everybody is doing it so that there should be a reason behind it. But okay, but yes, it's a good field, and I think by the end of, usually I offer 5 courses, 5 classes, but this time actually I'm not feeling that energetic. I thought, initially I was thinking to discontinue, I do not offer this course anymore once I was in the entrepreneurship, but then I learned that too many people have enrolled, so I thought, okay, I should just go ahead and do it. So I have divided all the exercises into 3 major classes. And the good thing about the bioinformatics, and I'll let you know in the last class, that you can have your own startup company. Right? You can have your own startup company where you can offer scientists, whether they're academic scientists or the industrial scientists, computational modeling experiments. Right? And I will give you an example also when I teach you about the docking, that when I was making the lectures for these exercises, and I was looking for which are these startups are already available and they are using molecular docking and computational type of experiments to offer to the scientist. And I went and I found some companies, and usually on the bottom of the site where you can communicate with them, you do the check-in with them to help desk or so. So I messaged them and I asked them that I want some docking help. They say, okay, yeah, we can do it. Immediately somebody responded. And they asked me how many targets you have and how many ligands you have. I said, I have just one target protein and one ligand. And then I asked, how much do you charge? So he did some calculation. There was a pause for 5-6 minutes. And then he said, we charge $1,000 plus 30% HST. And I will teach you that how long it takes. It takes hardly 15-20 minutes of your time to do that one target, one ligand docking. And they were, like for that 15-20 minutes, they were charging me 1,000 USD. Right? It's a lucrative thing, but this is here I can only give you some ideas, right? It's up to you how much you want to polish it, how much you want to learn more than what I teach you, and then decide whether you want to have your own startup company or not. And the reason behind having these bioinformatic exercises was when I was teaching to a program in the University of England, Masters in Medical Biotechnology, and they were all international students I was teaching. So I was teaching students from Iraq also. The students were coming from all over the world to get some Canadian experience. But then what happened due to COVID? They were all confined to their homes and hostels where they were living. They were paying 3 times the tuition fee than the domestic student for the Canadian experience. But they were not getting that Canadian experience because of the COVID, unfortunately. And at that time, when I was teaching them, I find they were very disappointed, very disheartened. And at that time, when I thought of them, why not I just design, because they cannot come to the lab and have hands-on experience, why not I design some computational experiments and then, give them some sort of a skill set so that they should not feel like a loser by the time they are graduated. And that's from there this idea of bioinformatics and computational experiments stuck to me. And then I wrote a grant application, submitted a grant application to different agencies, and I got funding, and from that funding I hired a student from computer science department and others, and then they helped me design these exercises. If you ask me, as such, I don't have any background in bioinformatics. So this is also, and now I'm teaching bioinformatics to the master's students. Not here also, in North American universities as well. So this also gives you an example that, and I have no degree, no background in bioinformatics. My background is in biochemistry, basically, right? And my specialization in neuroscience. I myself did not study bioinformatics, but I designed wrote a grant application, I got the funding, and I'm teaching this to the student. So this also gives you a very good idea about that you can learn any time in your life, right? And you don't need to be an expert, super expert to start doing something, to start teaching something, right? And I just learned at that time of my, which was not my background, no degree, nothing, but I realized the importance of that discipline. I learned it at their age, and not only learn, I come up with a novel idea. That's why they were able to find innovation in my idea, and then they give me money and ask, okay, yeah, go ahead, design the exercise and teach our students, right? So you can learn anything anytime. If you are stuck for whatever reason to doing something that you don't want to do, right? But later in your life you can change your field. I was also stuck, like some of you are saying, that you want to do PhD with somebody else, but you are forced to do PhD with somebody else, right? I was also stuck working on the enzyme inhibition. That was the only thing happening in the institute at that time. But as soon as I get the opportunity, I change it to neuroscience, right? And even after that, when I get the opportunity, I change my discipline completely. I am teaching now on bioinformatics, and I am not teaching entrepreneurship. It's something I never learned myself, right? So if you are stuck in a situation, don't victimize yourself by saying that, oh, I can't do anything. As soon as you get an opportunity, keep that spark going, and when you get the opportunity, go and practice what you want to do in your life, right? So bioinformatics, we'll move on, and I'll teach you what things that comes under the discipline. So you know what happened actually, all of a sudden, you will realize that all of us, we have become a data, right? All of us, we have become a data. Perhaps you might have heard news many times, a breach of privacy data, our data has been leaked, the piracy attack, the IT is under attack, and so on. So the data of, it's been everybody now has become data. If you are opening a YouTube channel, and if you are watching that YouTube, you are opening or somebody has opened a YouTube channel and you are watching a YouTube video, so how many times you are watching that YouTube video, it has become a data. So you have become a data, and based on how many people are watching, there are people, the content creators are making money out of it, right? So your, whatever you are doing online has become so much important right now. And there's so much data is being generated every day, not only in the life sciences, but everywhere. I've given you some of the examples in here, that the speed with which the data is being generated, it is not being analyzed, right? The speed with which data is being, see, look at here, in 2025, they say it will reach 181 zettabytes globally. And if you don't know what zettabyte is, I have given a scale here. Right, the kilobytes, megabyte, gigabyte, and this is a zettabyte. The next only available unit is yottabyte, right? And this 270 GB of healthcare and life science in 2020. So this is an old stats. And based on that old stats, how much data being generated every day, and this data is being contributed by, every time you go to Google and search something, whatever that you are searching, that information is being stored somewhere, right? Because once you search something, next time again you write something similar, you'll see that it's showing your previous research, search keywords already, right? So it's being stored and saved somewhere. Somebody is keeping record on your activity, your online presence, whatever you're doing, right? So all these data is being saved through social media. When you're going and shopping, making, ordering things using different buying outlets, and so communication, you're communicating through WhatsApp, TikTok, Insta, all that information is being saved somewhere. The emails, what is Internet of Things, any idea? Internet of Things. Internet of Things is what happened, for example, I left my university, it's 5 o'clock, I left, everything's done, I'm walking, driving towards my home. And starting from where I left in the garage of the university, where my car is parked, I can turn on the electricity of my house. I can turn on the heating or hot water or whatever. I can ask the vacuum, we have robotics vacuum cleaners. So I can turn it on so that before I come home and everything is clean. I can turn on my dishwasher, right? It's called Internet of Things. So all these things are connected through Internet. Right? This is called Internet of Things. So it's a big thing nowadays. You have your pictures in healthcare data also. Whenever you go to doctors, they're taking your lipid profile, right? They're taking your blood profile. They're checking your blood. Where this data is going to be stored somewhere. Similarly, if you're going to a hospital, imaging studies, ultrasound, MRI, CT scans, all these information data is being stored somewhere. So just imagine the amount of data that is being generated every day. That's why I'm saying it's reaching up to 181 zettabytes globally. Why? This is what I meant when I said that all of us, we have become data, like all of a sudden. So everybody has to transfer into the data. What is the life science research data, for example? The research that you're doing when you read the papers from PubMed, if you're doing some genomic and bioinformatic research, or for example, the project that the grant application I wrote for bioinformatics, I offered the course and I took survey responses from the students, if they like it or not. That survey response I published in a journal. Now I will show you the papers where I published the data. That data is being stored somewhere. It will always be stored somewhere on journal's website. Right? So this is how the data is being generated. The laboratory instrumentation data, the clinical trial data. Clinical trials are happening in, as I mentioned, clinicaltrial.gov.ca website. If you go to, you will find all the clinical trials have been done so far. Data is stored somewhere, right? So this is all the life science data. And as I said, the speed with which data is generated, it is not being analyzed with the same speed. What does it mean for you guys? When I say the data is being produced in life science or in general with the speed, but it is not being analyzed with the same speed. The data needs to be analyzed. Somebody is analyzing the data, but it is beyond their capacity because there's too much data being generated. When I say the data is generated, but it's not being analyzed, what does it mean for you guys?
Keeping check.
Sorry, what?
Keeping the data in check.
No, in terms of opportunities.
Yeah, opportunity. You can work on analyze these data that may not be analyzed yet.
So you have tons of job opportunities available. You have tons of job opportunities available in data analysis. Right? So this is what it means. The data, so much data being generated, we don't have enough people available to analyze the data. Right? So just imagine the potential of this field. And this is a very old stats I have from 2017 or so, just to tell students, how much money you can make with only a master's degree, 84,000 as a US dollars, and similarly 69,000 in epidemiology. And the good thing about bioinformatics is, if you're doing your master's in biochemistry, you have application in biochemistry field. If you are doing your master's in neuroscience, only neuroscience. If you are doing in organic chemistry, it's only organic chemistry. With bioinformatics, it has application everywhere in all the disciplines. In all the disciplines. And these are the examples I've given in here. Computational biology, okay, makes sense. Software engineering makes sense. Machine learning, but see in biostatistics, in mathematics, information systems, engineering, epidemiology, you just name it. So bioinformatics, once you learn how to analyze the data, you have these applications in so many places. This is how we used to do research. I don't know if still people are doing research this way. Does this one make sense? Very historic Googling. How do we used to do when there was no internet? When we started our PhD, this is how we used to do research in the libraries. Make sense? What is it? Any idea? What is he doing?
Library catalog.
Do we still have it?
Yeah.
No, but not in the HEC library, but do we have in the central library?
Yes, we have in library.
And people doing research this way? Still they are doing it? They're still living in that age? Some of your friends are saying they do. So, the thing is, like what we used to have, we go to the university, we didn't have any internet at that time, and I want to gather some information, papers about my field of study in my master's or my PhD. I have to go to the library. The library, they have these type of, small drawers or so, and the cards they are there, they are the size of a visiting card. And in that card, you can find the title of the paper, name of the author, the abstract, and the address of the author. That's all you can get information about, right? And then if you find that card that you have read, reading from the abstract is something, that the information that you want. So what we do, I need to know full paper, and how to get the full paper, I write a letter to the author. The address of the author in one of those cards. It takes at least, if everything works fine, at that time we have Mr. Mahmood Alam as the secretary of Dr. Tawarma. So we used to give him those cards and asking him, please post it. If he posts it immediately on the same day, it takes 15 days to get your card to the author in whichever part of the world is. And if that author is immediately very active, it is immediately post an article, put it in an envelope and post it to you, it takes the next more 15 days to get to you. In 1 month, we get the full access to the full article. This is how we did research. right? And then after you waste 1 month and amount of money for posting and getting back and forth, and after 1 month when you see the paper you realize, it's not what I want. And then just throw it. All that effort wasted, right? So this is how we used to do research. It's a prehistoric Googling, and now you have now everything is on your fingertips. You just write the paper, immediately get the article, you read it down, and you move on to the next one, right? So now how these, what is a database? How these databases were made? So the papers that we publish, as I mentioned, papers contain the, all the data, biological data, chemical data, imaging data, all that data is generated through as a part of research work. These scientists are doing all over the world. They are publishing their papers in different journals. So, like for example, we have 2.5 million quintillion data per day being produced. So here I'm showing you the research papers. These research papers are all over the different millions of journals. Right now we have millions of journals publishing these papers every day, right? So these, for example, if you're working on a protein, the name of the protein may be alpha-glucosidase, right? And that protein, you want to collect data about that protein, everything that has been published so far on the web, right? And that information is all over, everywhere, scattered in these papers. So what scientists did, initially from where the concept of database came in, they designed certain algorithm and software in order to put all that information in order, right? So what they did, they designed these algorithm and software to extract the relevant information, put them in the order, and design these bioinformatic databases. So the databases that we'll go through, some of them, I will show you how this information is curated and in the order, and just the way that you have put things in the proper catalogs or drawers or cupboards that you make. So this is how the information has been sorted into different categories, right? So these are the bioinformatic databases. This is done. Then some other scientists, so this is a raw data. We have all the data extracted from these papers and put them in different databases. For example, if you are working on alpha-glucosidase, so some of the way they have done it, like they have taken all the papers who are talking about only the structure of alpha-glucosidase in a different database, right? So it might be a structured database for alpha-glucosidase. Similarly, all the papers who are talking about only the sequence of alpha-glucosidase in different species, with mutation, without mutation, right? So that information has been extracted in a different database. So this is how the database was made. But this is all raw information, raw data. Then the other scientists, they design some more algorithm, they design some more tools, some more software to analyze that raw data, right? So when they analyze the raw data, they were able to do some sort of pattern recognition. What is the sequence similarity? Why certain sequences are similar? How the proteins are evolutionarily related? Alpha-glucosidase and how many species it is found, right? What is the relationship? What is the structural comparison? What is the sequence comparison? What are the functions? Are the same functions? So this is called the analysis. So from raw data, they analyzed and they were able to have all that information. So this is all built in the bioinformatic databases. So the databases that we go through, they are not simple database as when you do searches on Google and Google show you different websites. Those websites are very different than the bioinformatic databases that I will show you. So here, the raw data, so here you have all the raw data, it means that you have all the information in these papers. When you put them in the order, this is how you put the information curated in the proper catalogs in order. But the after analysis, what happened? You find the meaningful connections in between that raw data, right? So this was the raw information. You connected the points together, you make some sense out of it, and then finally when you analyze the data, you find a raw. This is the shape of a cat. Okay? So these databases, what are these databases? These databases need huge amount of effort, time and money, right? And it is not possible for one person to do it. Usually these databases are maintained by different institutions. Some of them are some local institutions, some are federal government controlling those databases. Sometimes some private companies have, it is a business model actually.
I did not show you in the entrepreneurship course. So some of the companies, what they are doing, they are just collecting all this information from different websites and creating those databases. And if you need access to those databases as scientists, they will charge you money for that, right? So it's all, again, it's a lucrative business for people to design the databases and then selling that data to the scientists, because it takes too much time, money, and effort to analyze the data. And these databases are coming; they are coming in all shapes and sizes: small, big, primary, secondary. For example, in here, there are some primary databases. Primary databases, they just contain the raw information. As I mentioned, the raw data is there in those databases. The secondary databases, they derive the information, for example, from the primary database. Here, we have protein database PDB. PDB, do you know what PDB database is? It contains the 3D structure of proteins, right? So this information it's not a raw data, it is the analyzed data, right? So this is called secondary data. And then we have, this is the data we'll go through ProSite, where we learn about more structural features of the protein. Similarly, some databases, they have, for example, this one in here is all metabolic pathway. If you want to know something about the protein that you're working upon, what are the metabolic pathways it is interacting with, that information, some gene expression database, and some more structured database. So unfortunately, I cannot go through all of the databases. I'll just go through some of the very basics one. And then once you learn how to, use those databases, then it's easy for you to learn and work on any database. Now, what is bioinformatics? If you ask me for a definition, it is the application of computational algorithm techniques. What is the problem with the indicator? Okay, the techniques, software, and IT. And okay, this application of computational algorithm, as I mentioned, some tools, some software, which are put together to do what? To store, organize, annotate, and retrieve the data from the research papers, as well as to visualize, analyze, or predict the sequence, structure, and function of proteins and genes. Because I offer this bioinformatics exercises as a part of a protein course, basically here I talk about only proteins, right? But if you want to talk about genomic data or some gene-related study that you're doing, there are other databases available. But because of I was restricted to the protein structure, so basically I talk about the protein structure. So what happened? These databases, they have built-in tools. You don't need to download a software. For example, when you're downloading a software to use it for something, the good thing about these databases, they have built-in tools in those databases. Sometimes you may need to download a software, which I will show you in the last exercise, for example, Chimera. And these databases, they have a command line also. So you just need to write a command the way you write command in the start prompt on the computer, or you use Linux or any other computer, and you write command. So these databases are built in to take your commands and then act accordingly, right? So these databases are not just ordinary website. Okay, so now we start the way I have designed them, because I usually teach them in 5 exercises. Here I will teach you in 3 exercises. So it's a complete project. How can you just use bioinformatics to from start to finish? And this is something usually people do in their MPhil or PhD, right? So here what I did, I will give you an unknown amino acid sequence. You would, using those databases, you will try to find out these sequences similar to your sequence. It's called sequence similarities. And then you will do phylogenetic analysis, and MSA mean multiple sequence alignment, right? So the database will do it. You have just unknown, that's all the information you have, that you have find out unknown sequence from somewhere. Now you're trying to find out the identity of that sequence. With which protein the sequence belong to, what are the evolutionary partners, what is the structure of that protein, right? This is a complete project. Unknown sequence you have, you do sequence similarities, phylogenetic analysis, multiple sequence alignment, and through this you will find out the structure of the protein. You'll do the structure determination by homology, modeling, and other things I'll show you. And finally, once you know the protein, and you find out the protein is responsible for a physiological condition. Physiological condition means it is responsible for a disease, right? So now the next objective is to block the function of that protein, right? And how can you block? By finding a ligand or a drug against that protein, right? So this is called the molecular docking. and through molecular docking we find the ligands. These ligands are actually the drugs that is being marketed, right? I don't know if I have the example or not, but maybe in the last lecture I'll show you that you following this scheme, these sequence of steps, there are many drugs in the market. It's not I'm just telling you imaginary thing. Many drugs that we have in the market right now, they were discovered through this process, right? And in the last lecture when I talk about the homology modeling, I'll give you the examples of those drugs. So this is called forward proteomics, because as I said, it's a part of this is a course I teach, BIOC 8740, protein structure function. And this is a course where I teach bioinformatics. I have taken the half of the slide out. Over there I teach them exactly the same thing that I teach them in this course. The next course I teach is called Biotechnology Laboratory course. So whatever they have done computationally in this course, they do exactly the same thing in the laboratory, in a different course that I teach again in summer, right? And in this way they find exactly same results, right? So this is how they learn that whatever they are doing computationally, they can also reproduce the same data in the lab, right? So this is how actually the drugs were discovered using computational analysis. Okay, so usually I have a theme for all of my courses. The theme I use for this course is called the bioinformatics treasure hunting. Why treasure hunting? Because as you are walking in a desert and you find this jewel, and then you realize, oh I see this jewel, there should be a big treasure hidden somewhere, right? So similarly, this jewel is actually the unknown amino acid sequence I'll give you, and then from there you have to do the treasure hunting. You have to find the entire treasure that from there this sequence belongs to. So this is called treasure hunting, right? So with just one small piece of information, 50 amino acid sequence, and then by the end you are done, you will get all the information above. These are the exercises I teach to the students. The very first one, where I give the unknown amino acid sequence, and these are the databases we go to. I'm not teaching this one in here because it might be repetitive. Then the multiple sequence alignment and phylogenetic analysis, and afterwards here we find the ligands for your protein. Ligands mean the drug component that they bind to, and these ligands you don't have to design your own unless you want to. You can also design your own ligand, but these we also have a database which has information about all these compounds. Right? So what you have to do, you have to just from that database, it's called Campbell database, need to get the structure of any compound and then dock your protein against that compound. Right? The last one is about the Swiss modeling and the chimera homology modeling and the docking. And in the end I ask, these are all enough steps to give you enough data to write a manuscript. And I have seen many of my students, I ask them, because protein you can find out, and they were interested. I said you can design your own compound, right? Using those, which is software. Which software we use to design a compound? You can design your own any structure compound or novel compound, and you can find out whether the compound that you design is novel or not. So design any simple drug-like novel compound and dock that compound against your protein of interest, the protein that you find out your sequence belongs to, and that's all you need to write a paper, right? So you can write a manuscript by the end of it. Okay, so this is because I generated this bioinformatic exercise as a part of the grant, and they asked me to create it as open educational resource. Open educational resource means I need to dump that in somewhere on a website so that the people all over the world have access to it, right? That was the purpose for it. So here is a website for it. If you go to that website, you will find this the entire exercises with the assessments, with the assignments, lab reports, everything I have dumped in that website. And these are the YouTube also link. I also have, actually I have the, whatever I'm showing you here, how to go and retrieve information from the databases. I already have made videos on that also, demonstration videos. So if you just watch the video, it shows you, go to this database, click on this, and get this information. So all that information is there on these YouTube links, right? So here already you have seen, okay, this is when I had only 22 downloads. Now if you go right now, you will see that close to 1,000 downloads. So this is something you can also link with your LinkedIn profile. because you are taking this course. So if you want your employers to know that you have taken this course and what you have taken as a part of this course, if you can link that to your LinkedIn profile also. So your employers can link on it and then click on it, and then they will be able to see what you did learn. Okay, and also yes, so I have also published papers on it. So in case if you want to know, in those papers I have already uploaded the entire exercises, the YouTube links and everything. So these are the papers I have published. This one and I think one more. Oh, this is my student. I just brag about it because I teach the student, and from one of the students I had take the help, and she was one of the co-authors. This second paper, right? So if you go to these papers, these are the links over there, and you can get more information about those papers. Okay, so now we start the very first exercise, identify an unknown protein with a unique amino acid sequence. Okay, so I am using a sequence, but I want you to use your own sequences. How? When we come to that one, I'll explain. So usually I start with this is the guy. So a person showed up to the University of Bethesda Hospital, and he was having chronic abdominal pain. And he has gone to the physicians with complaint of this pain. They thought initially, because when you have a chronic abdominal pain, usually people think it's ulcer, right? So they prescribe him certain anti-ulcer medicines, for example, omeprazole. It didn't help. Then they gave him some antibiotics. It didn't help, right? And they tried to change the antibiotics, but it didn't help. So then finally they thought when you couldn't find out anything, they finally decided to do a biopsy of that patient. And this is a procedure for the biopsy. So I show this to the students because when I teach them the laboratory course, in laboratory course they get this sample. They get this biopsy sample and then do exactly just to replicate the data that we get generate from here. So they took the biopsy sample, and then what they did, divided in 3 parts. In part 1 they find out, so from this biopsy sample, they grew the biopsy sample in the lab, and they try to isolate and see what is in that biopsy. So they isolated certain peptides. There were certain tiny peptides were in his stomach. Isolated them, identified. The second step part was to identify the peptides, and third part was to identify a ligand against those peptides, right? So that was the objective, the reason they took the biopsy sample. Biopsy sample they isolated, and then they find out different tiny peptides. They identify the peptides, right? Second is they try to, once they find out this is something that needs to be blocked, so they try to find out the drug against it. So this is what we will be doing actually. So exercise 1 is about that one. This one in here, and then exercise 2, and then exercise 5. So what happened, then you get the biopsy sample, grew the samples in the lab to have enough of the starting material. We lysed the cells, did the centrifugation, applied some chromatography technique, electrophoresis, and the protein sequencing and mass spectrometry to finally get the sequence of all the peptides which were found in the biopsy of that patient. Right? So these are the techniques that you have to go through. These are the techniques I teach in the laboratory course, right? But here I am telling you that how we get access to those sequence. So for me, the demonstration purposes, this is a sequence I'll be using, right? These are the 50 amino acid sequence that I'm using. For you, when you do your exercise, the bioinformatics, all the exercises I'll show you as a lab report, you'll be using your own sequence, and that sequence, you all have access to the Google Drive? Yes. I doubt the ones, I think the new ones. No, everybody do you have access to the ones who are taking the bioinformatics only? No. Okay, so it is actually the same Google Drive. I will ask Dr. Nidhi. Huh?
Yeah, it's in the previous drive.
No, but the student who did not attend the last class, they don't, right? So they don't have. But please contact Dr. Nidhi. I will also ask him to share the Google Drive with the students, the ones who just enrolled for this course. And or you can directly contact him. So what you have to do, so like the page that she is showing, you will find there tons of sequences on each line. So just choose any sequence and strike it out. Striking out so that other people should not use the same sequence. So all of you should have your own individual sequence if you want to do this lab report exercise. I don't want one person using the same, 2 people using the same sequence. So all of you should be using only one sequence, and once you chosen the sequence, strike it out, right? Don't write your name in front of the sequence. Because you will be evaluating each other's lab report. So if you know that whose lab report is it, then there is no purpose. Because I want it to be done anonymously, right? If I want her to analyze peer evaluation or evaluating her lab report, and similarly she will evaluate her lab report, right? But it has to be anonymous. Anonymous means they should not know whose lab report they are looking at it, right? So that's why just strike it out, don't write your name. Okay, so these are the databases that I'll be going through today. Unfortunately, as I said, I cannot go through all the databases. The very first one is called UniProt database. As the name suggests, it has all the information about all the proteins that have been curated in that database. So you will have information about the protein functions, domains, structure, modification. The good thing about these databases are they are just like, you know, the highway on a very busy Toronto highway. I usually give the example. So all these highways are interconnected, right? So from one road you can go to another one, and from another you can come back. So these databases are all connected. So you, from one database you can jump to another database, and then from that database you can come back to the previous one. So we have UniProt, which has all the information about proteins. PDB is the 3D structure data of proteins, right? The protein database. String database tells you about how the proteins, different proteins interact with each other. What is the connection between them, right? In case if it is something required by your project. And then we have SMR, Swiss Model Repository. It is the 3D protein structure. The difference here is 3D structure generated by basically X-ray crystallography, right? And the models, the structure that you have here is generated by Swiss model homology modeling or something, right? So it is computationally generated model, and this is experimentally generated models. Similarly, there is website ProSite. If you want to know more about detail about the structure of your protein, domains, families, functional sites, and so, then the ProSite is the one that you should use. So here, for the UniProt database, this is a link for the UniProt database. When you copy this in a new tab, something like this will show up over there. So what you have to do, you have to write the protein sequence, copy protein sequence in here. This one is off. So just follow. You see this? Follow the instruction. So you have to go to the link, copy your unknown amino acid sequence in the box. In advanced parameters, choose on here. For example, it is off. By default, you just make it on, and then hit run. When you hit run, it takes some time to do the search for you. Once the search is completed, it shows completed. Click on completed. When you click on completed, something like this will appear here. So here, within few seconds, you will find out the identity of your amino acid sequence, right? But it will show you a number of hits. It is up to you. You have to decide, because when you are working on a sequence, you have to go through and then see which one you think might be the protein that perhaps is your identity of the protein. Your sequences has been taken from that protein, right? So here I have given many examples. If you just click on complete, just something will appear, right? Click on first entry number, then sequence and isoforms on the left on the next page. So click on all these options one by one. This one in here, entry name and the organism. The more you click, the more information you find. So basically, from these databases, you just copy the amino acid sequence and you're trying to gather as much information as possible about your amino acid sequence, right? And this is what called the treasure hunt. We have the only one sequence available, but in just one step, you have access to tons of things. What do you have to do for your lab report? You need to write down the name of the top protein, closely matched sequence, what is the UniProt ID of that matched sequence, what is the UniProt ID that you can find in here about that sequence, what is the partial or original sequence of matched protein, what is the name of the organism in which it is found. For example, in my case, it is found in Helicobacter pylori. The sequence I have, it is from the Helicobacter pylori bacteria, right? So all this information, this is how you have to complete your lab report. Basically, you have to just go through all the pages, find out follow the instructions, look at the questions. Usually what I do, I share with the students a sample lab report. And this is my lab report looks like. Then I use my amino acid sequence. So it helps the student to find out exactly where to find their information about their sequence or their amino acid, right? So here I have again mentioned the same thing, that you go to advanced search and turn this off to on. In case, okay, sometimes what happens, because I'm using a bacterial sequence, yes?
Excel sheet
It's not there, then where is it? Where did you find it?
it is in the simple Excel format, so we cannot, if we strike, so we have to download it. It's view only format.
Oh, okay. Okay, so what needs to be done? Should be editable format or what?
No.
Okay, I will ask Fasi. So maybe just email Fasi or Dr. Nadeem and ask you that if they can give you access to, so that you can edit it. What about other files? What about other PowerPoints and PDF files?
They are,
Can you download them?
yeah, we can download them, but we cannot strike. You, as you were saying, any.
Okay, yeah, because I think it needs to be done online so that everybody can see, right? Okay, I will ask Fasi. Remind me, or if I forget in case, so you just email him and ask him that, if he can make it available in the editable format on the website, on the Google Drive. Okay, and if you want to know that what happens if there is a mutation is induced in your sequence, right? So now you know how to have access to these databases, the link that you already know. It's up to you how much you want to play with it, right? But you can just go and see, okay, what if the sequence is changed of my amino acid, order is changed of the amino acid? What if a mutation is induced? Will I get exactly the same protein as I have got before, right? So here I'm giving an example. So what I'm asking you, just change A with G in your protein sequence. What is A? Stands for? Huh? And G? Guanine? In proteins, how can you get in protein? Guanine is in protein that you will find. Where you find guanine? What is G? Oh my God! G is for glutamine? Glycine. For glutamic acid, what is it? Huh? Oh my God! Glutamic acid, I think is E or something. It's not GLU. GLU is when you use three-word identification. The G for glycine, A for alanine, guanine is in DNA, guys, not in protein. The very basics. Okay, and then what else? What is aspartic acid? Huh? Sorry? D. Aspartic acid is D. What about tryptophan? Huh? W tryptophan, what is Q? Glutamine? Q is glutamine? Sure. Tryptophan is W and Q is glutamine, aspartic acid is D, glycine is G, tyrosine? Tyrosine is Y, right? So yes, so you can just play with your sequence. So just change all the A's with G's. In case if you don't have A, just play with any other amino acid in your sequence, right? And then try to do exactly repeat the steps and see if you get exactly similar results or not, right? So I'm just asking, is your first match the same protein as before? If not, where is your previous match protein? So maybe now your protein, the one that you find out in the last step, is not showing up at all. So see, just one amino acid sequence, amino acid is mutated, and the identity of your protein is changed completely. Why? Because the structure of protein is changed completely, and the function of the protein is changed completely. It's very important that you preserve that information and then work with it. And did I explain this in the alignment? So here, let's see if I have it in the next one. So here what you see all these things, and the identity is called percent identity, the gaps, similarly some bit score, E values. Let me move on to the next one and I'll show you that how to interpret it and what does it mean actually. For example, you see the bit score in here. Bit score, expect value is also called E value, right? I will show you on the next one. The proteins with Campbell ID. Okay, so here, because I asked the student to, so this is how it shows up. When you write a copy of amino acid sequence, it is showing up that the system thing, the database thing, that your sequence perhaps from this protein, from this, perhaps from this, perhaps from this, perhaps from this, right? So it's just telling you that it might be from one of those sequences. Now it's up to you. Just go and open up each protein and you'll see that based on what is the source of your sequence. Where did you get the sequence? Your prof has given you some idea about that's this sequence I have gotten from a biopsy sample or from a poison or from an antibody or whatever, right? So based on whatever prior information you have a little bit available, see which protein that you think that perhaps might be your sequence belong to. Sometimes you have no idea. You have no idea what the sequence belong to. But you keep that information, write it down somewhere, or copy it, or save it, right? And then as we move on, you will get more information. It will give you more definite idea that the sequence that your protein belongs to from that particular protein, right? So here I ask the students, because I ask them to use the same sequence or the same protein throughout for all the exercises. So whatever protein you identify as you think that your sequence belong to, I ask them that use that same protein for all the lab report or exercises. In that case, I use the same protein which has a Campbell ID also. Campbell ID means one of the proteins should have a ligand against it known already and curated in the Campbell database, right? So in that case, I'm asking them here, just open these proteins one by one and check which of these proteins has a Campbell ID. Right? So Campbell ID means a ligand is reported against them. Note down those entries number that have Campbell or Amble IDs. Because sometimes it becomes problematic when I ask them to do the fourth exercise with the Campbell database and they say, ma'am, I don't have any Campbell ID. So I ask them here that just only choose the protein which has a Campbell ID. But in the sequences I have on the Excel sheet, all of them they have Campbell ID because I made sure afterwards. Okay, then click on first entry and then similar proteins. So when you click on the entry, the first one, something a web page like this will pop up and then you see the similar proteins. So if you click on the similar proteins, this section shows whether the protein you found in previous step has any known interactions with other proteins from other species. Right? So now you're, as I said, you're gathering information about your amino acid sequence. So when you click on each entry one by one, if you click on similar proteins, so you will find out whether you have some other proteins similar to the sequence that you already have selected or not, right? So here you're just trying to gather as much information as you want. Similarly, if you want to know the function of the protein, click on the function. This is called the shortcut menu, right? In all the websites, perhaps you notice when you load the page, it has a shortcut menu. Either you scroll down manually or you just click on it and it will directly take you to the place. So here you can find out the function of your protein also, right? Not only function, the mechanism of action, right? If it is an enzyme, your amino acid was an enzyme, this is the EC number for the enzyme, right? And the source of the enzyme, the reaction that it catalyzes, right? This is a catalytic activity. Because the protein that I have chosen, the sequence I have, it is taken from an enzyme, urease. It is a sequence of the urease enzyme, and this is the EC number, the enzyme commission number for the urease, and this is a reaction it catalyzes, right? So here, just only single command with single database. See how much information you are collecting about your protein. Similarly, you can go to the SMR, which is Swiss Model Repository. SMR, when you click, Control F to find SMR somewhere, and then click on it. This is the link. It will open a page in the Swiss Model Repository here. Doesn't show. So this is the structure of the protein, my urease protein, and there is more information about the experimental structure of that protein have been reported by different scientists in different papers, in different databases. If you want to know more about it, click on one of them one by one, and then you'll get more information about it, right? So this is called the Swiss Model Repository or SMR information. What you have to do once you reach on that page, find answer to these questions. Write down the PDB ID of match protein and how many experimental structure of your protein you find. For example, I find 4 structures here, right? So how many you are finding? So this is where I make sure that the students are going to the databases and looking for the information they are supposed to, right? So similarly, you will also find the PDB ID of your protein in the PDB database, how your protein structure is reported. right? So if I click on mine was 1E9, why? It is the PDB ID of my protein of interest, the protein structure. So it will take you a different database, PDB database in here, right? And here I ask the students that if you go here and then download the structure of your protein in the PDB format, because this is structure of protein we need for the docking step, right? So just go and download PDB format. I will explain this in that exercise also. But again, see, from the UniProt database, you went to a different database, Swiss model, which is the homology modeling structure or the experimental structure, and then you also went from there to another PDB, another database, which is a PDB database, and here you get more information. So it has much more than what you're seeing in here. It also can give you information about the mutation in the proteins, how many mutations are reported. Like if you just scroll down, and you know the good thing about the PDB, this is my favorite database. This database here, you can animate the structure of your protein. You can go upside down, left, right. You know, if you want to see particularly where the mutation is located, you can see through this annotated structures. Okay, what is BLAST research? Have you anybody who ever working in bioinformatics? Did your profs ask you ever that go and blast this sequence? Yes. What is BLAST? Sequence what? Sequence similarity. But then why don't they call it go and do the sequence similarity? Why do they call it BLAST? BLAST.
Basic local alignment.
So what does it mean? Basic local alignment tool.
There are two types of alignment based local alignment and global alignment. Local alignment is if your protein consists of 100 amino acids. So your protein will, you can search the protein that consists of almost 100 or plus 1 to 3 amino acids. But in the local alignment, you can search motifs and domains and different, short.
Less more
sequences.
focused.
Yes, more focused.
So, but then BLAST is more local or is global?
It's local.
So BLAST is local, right? And in BLAST we have certain other criteria that we look at. It's a bit score we look at, right? We look at the E score. What is E score? Or bit score? Any idea? No idea? You just know global and local about the BLAST? Okay, but anyhow, so what we do in order to do BLAST, okay, so that's what we have done with the UniProt database. UniProt is done. I showed you how much information you can get from just UniProt, right? And how many databases you can go to from UniProt. Now here we are doing a BLAST search. BLAST search means we are trying to find. So far we just posted copied the sequence of our amino acid UniProt, and UniProt using its own search engine letting us know these are the proteins I find perhaps your sequence belong to. BLAST is more related to just focus on the sequence, right? In UniProt database, sometimes you can get some noise also. You know, it's just roughly just comparing your sequence with the available proteins without much background search about the evolutionary relationship with your sequence and the one that they are proposing, the structural relationship between your sequence and the one that the UniProt is proposing, right? But BLAST is very specified, right? In BLAST, they're not only taking your sequence, the one that you're searching, they're also doing background research. In background mean, if they are saying that if your sequence is similar to some other protein sequence, in the background, suppose the one protein consists of 1000 amino acid and your sequence of protein belongs to a protein which is consisting only 200 amino acid sequence, right? So in that case, BLAST is looking at the sequence, the 50 amino acid sequence with this protein and that protein, as well as the background, the 1000 amino acid that belong to that protein and 200 amino acid belong to the other protein. Whether they are also similar to each other or not, they are evolutionarily conserved or not, the structures are similar or not, the sequence, further sequence is similar or not. So if there is too much noise, the E value would be very high, right? Too much noise means that your proteins, our sequence are similar, but it's something else which is a huge part of the structure of both the proteins, they are not similar to each other. This means there's too much noise. In that case, if your E score is very high, the chances of these two proteins are similar is very little. Right? So you have to have a very low noise ratio in order to for sure say with certainty that yes, this sequence that I have find out, it belongs to for sure for this protein. right? So the E score has to be smaller. I explain the E score even on the next one. The BLAST tells about bit score, E values, whereas the step 0 of UniProt search tells only the match protein. So the UniProt does not look at the bit score, does not look at the E value. Only BLAST that looks at the E value and as well as the bit score to give you more specified and highly predicted and more similar and authentic data as compared to just general UniProt search, right? So here I have given the example, for example, here. So, these are the proteins it find out similar to my sequence. All are urease subunits, right? The different species reported by different scientists in different papers. Sometimes they are similar species, sometimes they are derived from different species of, the urease enzyme, for example, is produced by many bacteria, right? So if these sequences reported by different scientists, they're isolated from different bacteria. So now through bit score and E value, you will come to know if those bacterial enzymes or the enzymes from different bacterial sources are evolutionarily related to each other or not, right? So if you have a high E score value, it means they are not, right? If you have a low E value with a little background, it means they are, right? So this is how they have given you the E value, the percent identity, and the maximum score, right? So I have an example on the next one. So E value, as I mentioned, E values higher than 0.001 are generally considered unreliable. So for E value, it has to be very small, very tiny, like 0.00001. That is better. This means that you're very likely that your sequence belongs to the protein that you have identified as the protein from where you isolated the sequence. Similarly, on the other hand, the bit score is a rescaled version of raw alignment score, and it has to be higher. The higher the bit score is, the more similar the proteins are, the sequence that you have find out. And percent identity is very simple. Percent identity means if out of 75 amino acids, 50 amino acids are similar in two proteins, what is the percent identity? Can anybody calculate? Out of 75 amino acids, both proteins have 75 amino acids, 50 are same in both of them. What is the percent identity? 75%?
66%.
66%? Yes, something similar. But now I want you to please find out the E value. So the question is, you can find out E value, that there is a formula for it. This is a formula for the E value. M, N and 2. What is M? Assume that the query sequence, this is your sequence that I gave you, M with a length of 100 has been compared against a sequence N with a length of 150 and the obtained bit score is 28. What is the expected E value? Expected E value or E value is the same thing. So this is a formula for expected E value. M is the query length of 100. N is the length of 150 of the given amino acid. And S is the bit score, right? So what I have done here for you guys, I already have replaced the values and I already also simplified it for you. Can you let me know the answer E? What is E? 0 point? How many zeros? 40. Are you sure? Did you do the calculation or are you just guessing it? Where is your calculator? I don't see it. Oh, you did it already?
Minus 28, it will be minus.
So it is, yes, 0.00558. So this is the E value, which is telling us that the smaller the noise, the more similar the proteins are. Right? This might not be I think for some of you perhaps might sound is complicated. Unless you're doing research in this field and then you're working on a project, it makes more sense, right? And unfortunately, I cannot go more simpler than that. Alignment on UniProt. Okay, so that was one way of finding out. First, we tried to find out the amino acid sequence in the UniProt database. Second, we tried to do the BLAST search to find out the sequence similarity between the amino acid sequence and the existing protein. And the last one is called alignment. It is something like multiple sequence alignment. So here, what the database does, it just forget everything else. All the information about your protein, the structure, function, evolutionary relationships, related proteins, similar proteins that interact with. It just focus on the sequence, and it shows you the data in the real time. So here you have your sequence, and then if you go to the alignment, I mentioned the alignment section. Then how would you go to alignment? In the alignment, this is my sequence that I pasted, right? And this is a sequence that it find it's similar to, and it is very wild. Here the system is only comparing the sequence. That's it. Not doing anything more than that, right? So here the sequence comparison. So for the first protein, it did your sequence and my sequence. For the second protein, that my sequence, your mean like the given sequence that they think it might be similar to. It is also giving you E value here, the percent identity, right? And the gaps and the positives. So this is a different one more way of comparing your sequence with the available proteins, right? In the multiple sequence alignment, we exactly do the same thing, just comparing the sequence. We are not concerned about the structure, the function, the evolution or whatever, right? So here just giving the sequence of your protein and the protein that you find out and asking you can just make your own choice. If you think that this one is better, choose this protein. If you think that one is better, choose that protein. There are different parameters mentioned here, bit score, E values, percent identity. So if this is the criteria that you want to use to choose your protein of interest, you can go ahead and do it, right? So I have explained how the identities are done. For example, the same thing somebody was asking, when I was asking that, out of 54 amino acids, 42 amino acids are similar here. So it is actually 78% comparison between your sequence and the given protein. Okay, so this is yes. If there is a match with 100% amino acid identity to a protein in the database from the same species, then your protein is not novel. I remember the last time a student come and asked me, ma'am, I have found a protein and I want to know whether it's a novel protein or not. So here is the criteria. If your protein is having 100% your sequence, you find with 100% identity already existing in a database, which means your protein is not novel. Which means it's already reported by somebody already in one of the papers or research papers, journals or database, right? If the top match has less than 100% identity, then it is likely that your protein is novel. So when you copy your sequence in any of these databases, UniProt, BLAST, or the alignment what I mentioned, and you don't find 100% similarity. From top to bottom, it's like 96%, 97, 90, 80 something, but no 100%. It means that you most likely that your protein is a novel protein, because there is no protein exists in the database, any of the databases which has 100% similarity to your protein, right? But at the same time, if there is a match with 100% identity with your amino acid sequence, but to a different species, right? Your urease is from Helicobacter pylori bacteria, and you find 100% similarity with a different bacteria called Bacillus subtilis, right? It means that your protein basically is from a novel gene, right? So that in that case also you have found a novel gene. So if you have, again the question, let me just repeat again. So if you find that your sequence has 100% similarity, one of the existing database protein which is already there, there's nothing novel about it, right? But if you find out there is no 100% similarity in any of those, so your protein is a novel protein. It has not been reported in the literature anywhere by anybody. And again, if you find 100% identity, you still have a hope. But in a different species, your sequence that you have, it is from Helicobacter pylori, but it is having a similar sequence to a different bacteria from a different species. It means that both of them, they were evolved from a novel gene, right? Not the novel protein, but the gene was novel. So this is how you can find out. Okay, there is a one-story house in which everything is yellow. Yellow walls, yellow doors, yellow furniture. What color are the stairs? Sorry, what? Huh? But even one stories have stairs. This one has stairs here.
One story there is no need.
There is no need to yellow color?
No, there is no need to stairs.
No, but stairs are always there. I didn't see any house without stairs.
There is only one
So even coming on this stage, I have to, this is the one stage, stage, isn't it?
And these are steps, not small steps, dark yellow or brown.
Brown?
Yeah, because the first is brown.
Okay. What's your name?
Mesa, Victor Mesa.
Huh?
Victor Mesa.
Dr. Mesa is saying that it might be, because of the contrast, she might be brown or yellow color. Do you agree? No? So they don't agree with your color contrast. Maybe it is in Iran. What color contrast will you use here in Pakistan, in Karachi? White? White color contrast? White? Guys, quickly. White, yellow, brown, what? Green? Huh? He is still stuck on it. He is stubborn. Very stubborn. Saying no stairs. You are the only one who is saying that? How many people believe? Half of the class? Huh? One story, no stairs? Are you sure? Such a silly question, right? In one story there is no color, so it is yellow. Look at the same, but see, I am showing here. Sometimes you have these type of stairs, isn't it? Anyhow, okay, string database. This is a new database. So forget everything about Unicode that we learned, GSMR or the PDB or the BLAST. Now a new thing, right? So all depend, because all depend on your project. What is the objective of your project? What you're supposed to find out. Sometimes I find that your profs, they give you a protein sequence and they say go find out with which this protein interacts. That is your project about sometimes, right? The string database, what it tells you the proteins with which your protein interact. For example, my protein, when I wrote down here my protein name, the organism Helicobacter pylori, I did the search and I find this one in here. I click on Helicobacter pylori. This is the organism from where the urease alpha protein was taken, and this is what I find. Right? So when I click on that one, it is telling me these are the proteins with which your protein interact. Right? So if you're not interested in knowing the identity of your protein, the structure or function or whatever, you just want to know with how many proteins might interact, go to the string database, write down the name of your protein, choose the organism, and then here it is showing you the names of all the proteins with which my protein interact and what percentage interact, right? 99.9, 99, 96.8, 95.5, right? So it's just telling you that these are the proteins with which your protein, whatever function it does, interacts while doing its job, and this is the percentage of interaction. Right? And the last one is called domain finding. If, again, as I said, depending on your project, the stuff that you're doing for your MPhil or PhD, if your target is just to find out the functional sites on your protein, right? What are the functional sites? What are the active sites? Where are the binding sites? Right? More towards only structure of your protein. Then go to the cross-site database, and here you copy the sequence as I did in here. Click on Start the Scan, and it will give you all the information about how many domains are there in your sequence. And sometimes it predicts. So, for example, with 50 amino acids, you cannot know all this information. Right? You have only 50 amino acids. 50 amino acids are not enough to give you the structure or the functional sites or the binding sites, but you are asking the software to predict based on that information. So based on that information, you can suppose if you find out the novel protein, it's a novel protein and you want to know more about it, how can you find out more about it if it is a novel protein, right? So this cross-site database predicts that information for you, right? And sometimes, some people I have seen, they do their I don't know the MPhil or what, at least master thesis are like that you have a sequence and you find out it's a novel protein. Now you are just making a prediction and publishing a paper based on that. Because when somebody actually does the experiment and they exactly find out what you predict. Right? So it's a master thesis. It's just to work on this database and getting as much information as you can. Okay, so that was the last one. So it is so when you submit your assignments, please don't submit your assignments like this. Then I'll be having a hard time to find out who has done what. And all assignments should not look like same. This is one of the reasons I have given a separate amino acid sequence to all the students so that everybody does their own job. Right? Usually sometimes boys, one person works for everybody, and then they submit all the assignments. In that case, I have given the sequence separate to each student, and you will have your own protein, and then you have to gather all the information about your own protein of interest. Okay? And the last one is, okay, so everybody, this is the liberty I give students, because what happened when I have these type of exercise and I have sample lab report uploaded, students think that they have to exactly do what I'm mentioning here. Whatever searches I've shown you, whatever the screenshots I have shown you, it is regard to my amino acid sequence, the one I'm using, right? And that's why I'm getting those results. In your case, perhaps some of your amino acid sequence are from human resource, right? So from a different species. Perhaps you don't find the genome for it. Perhaps you don't find, there is no structure known for that protein, that sequence. So you don't have to follow mine. The main purpose of these exercises is to help you learn how to gather information from these databases, right? So whatever way you can find information about your protein of interest, just go ahead and do it. You don't have to stick to what I actually showed you. Purpose is just to learn and do your own way. See these ladies, they're just thinking about your own way. Somebody is jumping, somebody is going under, and the last one even does something very phenomenal. See, they are not even following each other. Now she's thinking, looking at him, he's too tall. What should we do? We're just thinking, having some conversation, mind conversation, and then she says, you know what? Let me help you. And, right? So the ultimate goal is just to cross that path, right? And whatever way you want to do, you can do it. Okay, guys? Any questions? So you have to have a submission. You have to work on the lab report and just follow the instructions. Questions are, I think I already uploaded this on the Google Drive. It's on Google Drive. Okay, let me upload it. I think I can do it, or I will ask first. So what you have to do, follow the instructions, find answers to the questions. It should not take more than, I would say, 15-20 minutes of your time just to work on one exercise. So once you submit it, and then that report will be evaluated by one of your peers. So far, so good? Any questions? Sorry? I didn't hear, sorry.
He's asking about the submission date of the time.
Every day.
No.
Usually, yeah, because I teach these exercises once a week, every Thursday. So I ask the student, I'm teaching on Thursday, so I ask them submit on Tuesday, the coming Tuesday, and submission and evaluation on the coming Thursday. So in this way, they have a week or so, right? Here, unfortunately, I'm not here for too long. But if you want me to do all the marking before I leave, then it's better to submit as soon as possible. Right? So it hardly will take 15-20 minutes of your time. You think you find it difficult or complicated? No. So you have to just go to the websites, copy your sequence, and then find out. And you just have, it's a one-word answer. You don't need to write down the questions also. Just, the question one, rather than writing the question, just number one and then your answer. So one-word answer that you have to provide. That's it. It's not like you have to write an assignment or something. Okay? We done very quickly actually, 1.5 hours. Any questions? Do you want, now you want 5 minutes break? No? Just you want 5 minutes break? I'm giving the break for the entire day. So we are done.
Excuse me, ma'am.
Sorry?
Excuse me, ma'am.
Yes.
How can we submit lab report?
How should we submit, guys? Any idea? Huh? Which are the forms available? Word file is good. Yes. What do you think? What do you suggest? How should I?
How can we submit lab report, ma'am?
He's really getting impatient. Guys, I need answers so I can tell him. So okay, I would. Can you send it the hard copy? Where are you?
I'm from Punjab. I'm from Punjab.
Punjab, you are just telling me that I'm from a different country. Which city in Punjab?
Multan, ma'am. In Multan.
Can you post it to us? print it out and then post it at the iccbs address. Is it possible?
Yes, ma'am, I can.